Hi everyone, I'm Frank Owens from Mississippi State University. I'm here today to talk about wood identification of Peruvian timbers using the field-deployable XyloTron system. This talk is about automated wood identification using computer vision. Let's start with a brief introduction to the co-authors. Prabu Ravindran from University of Wisconsin-Madison, Alex Wiedenhoeft from the Center of Wood Anatomy Research, USDA Forest Products Laboratory, Patricia Vega, Oregon State University, Rolando Montenegro from Universidad Nacional Agraria La Molina, and Rubin Shmulksy, Adam Wade, and myself from Mississippi State University. Funding for this research was provided in part by the following: U.S. State Department, Forest Stewardship Council, University of Wisconsin-Madison, and USDA ARS. Let's start by thinking about computer vision-based wood identification. But to do that, let's remind ourselves a bit about conventional wood ID. So conventional wood identification is human-based, and it's performed using a hand lens and/or microscope. It involves observation of anatomical features of the wood and then comparison of those features to reference descriptions or specimens. It requires substantial training in wood anatomy. We'll contrast this with computer vision-based wood ID, which involves computer analysis of digital images. For hardware, you need a digital camera, lens, and computer. For software, you need feature detection and classification algorithms, which are a kind of artificial intelligence. Computer vision-based wood ID requires little human training to operate a device, and minimal to no wood anatomy knowledge is required. I mentioned the XyloTron in the title of the presentation. What is a XyloTron? Well, a XyloTron is a computer vision-based system designed at the USDA Forest Products Laboratory. Alex Wiedenhoeft is one of the co-creators. It's open source and field deployable. For hardware, we use the xyloscope, and you can see what that looks like in the inside here. It contains a camera and a lens, and the xyloscope hooks to a laptop by cord. And for software, we use a convolutional neural network for identification. This is abbreviated CNN. I'll use this abbreviation throughout the presentation. How does a XyloTron identify wood? Well, first you have to train it. You start by deciding what kinds of wood you want the XyloTron to be able to identify, and then you gather lots of specimens for each of those types of wood. You prepare the specimen's transverse surface by sanding, and then you capture many images of each class. When I use class in this presentation, I'm referring to a type of wood that you want the XyloTron to identify. Usually this corresponds to a genus, but not necessarily so. Then you're going to designate the class membership of all of those images you took. So for example, if I'm doing North American hardwoods, I would take all of the pictures I took of red oak specimens, put them all together and say these are red oak. I would do the same for white oak, the same for cherry, etc. Then you're going to train the model by showing the model those images that you took and telling it what they are. This is a picture of red oak. This is a picture of ash. This is a picture of white oak. This is another picture of white oak. This is a picture of cherry, and so on. You'll evaluate the performance of the model, iterate as needed, and then finalize the deployment model. Once you've trained the XyloTron, how do you use it to identify a specimen? Well, first you prepare the specimen's transverse surface, then you place the xyloscope flush on the specimen, capture an image, wait a few seconds, and then read the output. The output is expressed as a confidence from 0 to 100% for each class. As you can see here in this output, it's 78% confident that it's white oak, 16% confident that it's red oak, 5% confident that it's white ash, and then it lumps everything else into that other category and assigns it 2%. It just so happens that this specimen was white oak, so the XyloTron's top prediction was correct. Now let's think about illegal logging in Peru. State-owned Amazon forest covers approximately 60% of Peru. Over 68% of logging concessions show illegal harvesting. At least 58% of exported lumber is illegal. These are 2016 statistics. Now this is obviously not good. but there are some challenges to prevention that revolve around this reliance on human-based wood identification expertise. There are limited opportunities to acquire such expertise, limited institutional forensic wood ID capacity, and mostly localized access to xylaria. I want to emphasize this is a problem not only in Peru, but in many other places as well. Now, one possible solution might be XyloTron. Extensive wood anatomy expertise is not required for deployment. It's field deployable. You can take this right out into the field with you. It has comparable or better accuracy than expensive spectrometric methods. It's already been field tested on African species in Ghana, and a model for Colombian woods already exists. So we can take all that experience from previous projects and use it to answer the challenge of creating a model for Peruvian woods. Here are the objectives of the project. Develop a CNN model on the XyloTron platform to identify important Peruvian commercial species, evaluate the model in a way that approximates field testing, and ready the model for on-the-ground deployment and evaluation. Now, here are some important considerations that separate our work from other work that's been published. First, we train the model with representative input images. This means that we minimize artifacts. Those are scratches, gouges, things that the XyloTron might interpret as features. And we also make sure to include sufficient variation. So that means we don't want to take all of our specimens out of the same board, for example. We want to evaluate on a specimen basis and not on an image basis, and I'll talk more about that later in the presentation. Finally, we want to test the model on specimens not involved in the training. This is very important for generalization. Materials. We selected 24 commercially important Peruvian timbers, representing 57% by volume of commercially harvested roundwood and 66% by volume of sawnwood. These are 2019 statistics. We have 24 classes, each containing anatomically indistinguishable taxa. If you look at this table on the right, you can see that most of these classes correspond to a genus, except for these two here, Brosimum A and Brosimum U, which can be anatomically distinguished from each other. Those taxa in Brosimum A are indistinguishable from Brosimum allicastrum, and those in Brosimum U are indistinguishable from Brosimum utile. Here are the xylaria from which we took the specimens that we used in the project. I'm not going to read these. You can go ahead and take a look. All of the red ones we used for training, and the green one, which is the David A. Cribbs Wood Collection, now at Mississippi State University, we used for testing. That xylarium is abbreviated PAC-W, and we will use that abbreviation throughout the presentation. Here is a summary table for the specimens. For our training data, we had 1,300 specimens. For the testing data, that's data from that PAC-W collection, we had 119 specimens. Number of images, 5,715 for the training data, 529 for the testing data. The number of taxa, 186 for the training data and 70 for the testing data. Please note that asterisk. It says Peruvian species plus macroscopically indistinguishable congeneric species native to South America. What that means is, even if the specimen was not from Peru, as long as it belonged to the same genus and it was native to Amazonian South America, we included it. Methods. Sample preparation. We polished the transverse surface of each specimen with a rotary sander using progressively finer grit up to 1500. Then we removed the dust with compressed air and adhesive tape. Image collection. We imaged the transverse surface of each specimen with the xyloscope. Each image captured a 6.35 millimeter square area of tissue. We took these in color, so we have RGB channels. and each image was 2048 by 2048 pixels. We captured multiple non-overlapping images per specimen. We did this because we didn't want to have large portions of the same area appearing in multiple images. Linux-based and Windows-based versions of the XyloTron software were used to collect reference images. At least 7 separate XyloTrons were used by 11 different operators. Here's our CNN architecture and training scheme. We'll start with figure A. You can see the architecture is made up of the ResNet 50 backbone and the custom head. We go down to figure B. We can see the details of the custom head. This is average pool, max pool, concatenation, batch norm dropout ReLU, batch norm dropout softmax. We use a cross entropy loss function. We kept the pre-trained rates in the custom head, in the backbone frozen. We set the weights in the custom head initially as random, and then we adjusted through training. In the second stage of training, we allowed fine-tuning across both the ResNet 50 backbone weights and the custom head rates. For training, we had a 24 class model. The input images were 2048 pixels by 2048 pixels. Out of each image, we sampled a 2048 by 768 strip and then downsampled that to 512 by 192. We fed the CNN in batches of 16. For data augmentation, we used horizontal and vertical flips, slight rotations, and cutouts. The hyperparameters were consistent across training runs. PyTorch and Scientific Python tools we used for model definition, training, and evaluation. Results. Part 1 of our model evaluation was a 5-fold cross-validation. We took all of our images and randomly divided them into 5 groups. Each of these groups we call a fold. Then we take 4 of those folds and we train the model. And then we have the 5th fold serve as the validation set. And then we do that 5 times so that each fold takes its turn as being the validation set, while the other 4 are used to train the model. We made sure that the images from the same specimens were always in the same fold. 39 out of 1,300 specimens were misclassified, which works out to 97% accuracy. The figure here on the right is a confusion matrix. Here are all of the names of the classes, and they're the exact same names here on this axis. The only difference is these are the classes that the specimens actually were, and these are the classes that the XyloTron predicted. So where you have this orange diagonal, these are all of the cases in which the XyloTron predicted the correct class, and all of these numbers out here and down here are instances where the XyloTron predicted an incorrect class. Part 2 of the model evaluation served as a surrogate for field testing. We took all of the training data across all 5 folds, and we used that to train the model. And then we used the model to classify specimens from the PacW xylarium, which was not used in training. The top prediction was correct for 103 out of 119 specimens. That works out to 86.5%. The correct taxon was in the top 2 predictions for 110 out of 119 specimens. That's 92.4%. Now you'll notice a drop in accuracy when compared to the 97% we got from the 5-fold cross-validation. Loss in accuracy when classifying new specimens has been seen in previous studies, including Ravindran et al. 2019. Now, this is likely due to several factors, including subtle differences in anatomical patterns that you'll see among specimens from xylarium to xylarium. Here's our model evaluation summary. Again, the 5-fold cross-validation had an accuracy of 97%. PacW specimen was in the top prediction, 86.5%, and PacW specimen was in the top 2 predictions, 92.4%. Discussion. Now let's take a closer look at misclassifications. It turns out that we can divide these misclassifications into 3 different types, each type representing about 1/3 of the total. In type 1, we have a case of anatomically typical test specimen being misclassified as an anatomically similar taxon. So image A is from the class Amburana, and it was mistakenly categorized as Ormosia. Now these two classes look very much alike, so this makes sense that a misclassification could occur in this case. Type 2 is a case where we have an atypical test specimen misclassified into a taxon it more closely resembles. So image C is kind of a strange instance of Virola being misclassified as Sweetinia. And it just so happens that that atypical case of Virola looks a lot like Sweetinia. So that type of misclassification is sensible as well. Now we get to the third type, where we have a typical test specimen, in this case of Cariniana, being misclassified into an anatomically disparate taxon, Cedralinga. Now these two classes, to my eyes, don't look anything alike at all. So this is a really, really interesting case of misclassification that is worthy of further study. Humans are likely to make type 1 and 2 misclassifications, but not type 3. And here are the takeaways from today's presentation. We created the largest tested computer vision wood identification model for Peruvian woods. We evaluated our model on a specimen level as opposed to an image level. We demonstrated generalization by running the model on new test specimens from a separate xylarium. And remember, we said that this was a useful surrogate for real-world field testing. And then finally, we demonstrated that the model appears to be robust to different operators and xylotrons. Next steps, we're going to take the PAC-W specimens and fold them into a new field model together with all the training data from the 5-fold cross-validation. We will test the new field model on specimens from Xylaria in Peru and then conduct real-world field testing along with independent forensic validation of specimens. Then we'll deploy the model for real-world use. So that's all I have for today. Thank you very much for your attention. I'll show you my image credits, and then if you have questions, I would be happy to field them. Thanks again.